Hello, everyone. My name is Harshu, and welcome to the session on safeguarding biotechnology from catastrophic risks. I'm very excited today to introduce you to Professor Kevin Esvelt. Esvelt is an assistant professor of the MIT Media Lab, where he leads the Sculpting Evolution Group in exploring evolutionary and ecological engineering. He received his Ph.D. from Harvard for inventing a synthetic microbial ecosystem to rapidly evolve useful biomolecules, and subsequently helped pioneer the development of CRISPR, a powerful new method of genome engineering. Esvelt is credited as the first to describe how CRISPR gene drives could be used to alter the traits of wild populations in an evolutionarily stable manner. He has worked to ensure that community discussions always precede and guide the development of technologies that will impact the shared environment. Today, Esvelt will be discussing why information hazards are more acute in biotech than in any other field, how cryptographic screening of synthetic DNA can help, and the ways in which a nucleic acid observatory could provide advance warning of any exponentially spreading biological agent. We invite you to use the swap card platform during this session. On the right-hand side panel, feel free to share your thoughts on the live discussion board and leave and upvote any questions for our speaker to answer at the end of the presentation. After the session, we invite you to use swap card to set up meetings with your fellow conference members, and Professor Esvelt will also be on GatherTown for a half hour following this presentation to answer any questions that we weren't able to get to. So please join virtually to continue the conversation. And without further ado, welcome Professor Esvelt.
Thank you for the very kind introduction. So I should start with a confession, which is that I didn't really intend to get into this particular field. It just so happened that my research, which focuses on engineering evolution and controlling the fitness of engineered constructs in the wild, is highly relevant to catastrophic risk because biology, in order to pose a risk, has to scale. We need to turn the microscopic into the macroscopic. And that requires what I think of as invasively exponential growth. And my confession is that this absolutely terrifies me. And that terror sits pretty uneasily with my conviction that in the field of ecotechnologies, where we're engineering the shared environment, something people can't opt out of, we need to be open and transparent and invite communities to tell us what to do. How do we square this concern for catastrophic misuse with the conviction that things go best when we're open and transparent about what we're intending to do in the laboratory? So to tell that story, I need to go back a little bit to CRISPR-based gene drive, because for a lot of people, it doesn't seem to make sense to even bring it up as a consideration. Even in the midst of a global pandemic, the notion that we could plausibly engineer something that would spread exponentially just doesn't seem plausible. But in fact, we do know that engineered invasively exponential spread is possible, at least in the laboratory. We can build CRISPR-based gene drive systems in organisms such as mosquitoes and others that will spread invasively through caged populations. We haven't done this in a wild population, true, but our models suggest that it is highly invasive. That is, here is a simulation that we made that of a very poor drive system. This is something that only copies itself half the time, and the rest of the time it makes a resistant allele that is going to ultimately outcompete the drive system in the population because it's not as costly. This is what you get if you just throw CRISPR and a germline specific, a reproductive cell specific promoter into fruit flies. And what happens is that the drive system will rise to about 30-something% of the population, and then it will fall. But if we plot this peak frequency of the drive system against the number of organisms typically released for a simulation, then what we see is that for most cases in which we introduce even a handful of organisms, the predominant outcome is that it invades the population and rises to its maximum frequency before getting outcompeted and falling. In other words, it doesn't take very many for something like this to take off. Now, I'm not actually concerned that much about potential misuse of CRISPR-based gene drive. I could be wrong, but we did decline to tell the world about it until over a year after we actually realized that it was possible. And specifically, the concern was over potential misuse. Because before, no one had even really thought of the possibility that a single researcher might be able to build something in the lab, let it go, and have it impact a whole species. I'm a huge science fiction nerd. I haven't seen anything like that in science fiction. And yet, gene drive is slow because it spreads over generations. It can't quite double every generation in the wild. It's obvious, at least if you look for it, because if you sequence the genome of an organism, or even metagenomes in the environment, you can see it. And it can be blocked. The very first thing we did when we tested the first one in yeast is we showed that we could build a drive system that would disrupt the gene, AID2 here on the top left. And we built, at the same time, in parallel, a reversal drive that would cut the first one and restore the function of the missing gene. And they both worked beautifully. So anything that is slow and obvious and easily blocked is just not much of a threat. I wish that all biology were like that, but we need only look out the window at the world today to see that it is unfortunately not. If we take SARS-CoV-2, for example, it is comparatively fast. It has a doubling time measured in days. And here's a happy little exponential growth plot of coronavirus deaths in the United States. It's, I'm not going to say one of the most subtle viruses, but it's more subtle than most in the sense that it does this asymptomatic and pre-symptomatic transmission thing. And also, it's really not amenable to mass testing just because RT-PCR tests are only 75% sensitive on day 6 post-infection. And it really doesn't get any better than that. So we can miss a lot of cases. And note that maximum contagiousness is typically on day 5 or day 6. So you're usually going to miss people. Fortunately, it does seem to be blockable. We have very, very good vaccines, but note that we've been in this pandemic for quite some time, and we're only now rolling out vaccines in the wealthiest countries. So when you put this together, it looks a lot like pandemic biology appears to favor offense. It seems to be the opposite of gene drive systems. That's worrisome in and of itself, but of course there's the possibility that other forms of biology could similarly favor offense. But let's stick with pandemics right now because it's what everyone else is focusing on. How bad is this? Well, this pandemic has claimed more victims than the number who would perish if the highest yielding operational intercontinental ballistic missile loaded with a fusion weapon were to detonate in the densest of the world's cities. In other words, the possibility that humans could engineer pandemics like this one is on par with nuclear weapons. We don't treat it that way when it comes to security risks. Even though we know that historical pandemics were actually much worse than the one that we've seen over the last couple of years. I don't want to dwell on the numbers. Some of these we could readily defend against today. Others we would still have some problems with. But all of these were worse. And yet, it's because of these that a lot of people say, well, Mother Nature is the worst bioterrorist. Humans couldn't possibly do better, right? We have a poor record of saying that something will never be accomplished. After all, the Wright brothers famously stated that flight was decades away, and then they did it 2 years later. Fermi said, no, fission is far away, and then 4 years later, he was overseeing the first reactor. Mother Nature is not the ultimate bioterrorist, and Chris Bakerly has an amazing perspective in Stat News on this, making the case. And he says, you know, if making a bioweapon is like a competitive soccer match, or football for all the non-Americans out there, then Mother Nature is the equivalent of an especially persistent horde of 5-year-olds, all squabbling over the ball and unable to do more than one thing at the same time. But I think it's actually even more extreme than that. Mother Nature is like a tremendous horde of 5-year-olds fighting over the ball who have no idea that there are goals, much less that the objective of the game is to get the ball into the goal. Humans, on the other hand, are perfectly capable of seeing what the goal is and engineering something to match. The challenge is, how are we going to defend against this when we know that today we cannot at our current technology level, defend adequately against a mild pandemic? Is it likely that we will learn how to make natural class pandemics? Well, do you know people who are seeking to learn how to predict which viruses would cause a pandemic upon spilling over into humans? Well, yes, there's a project Spillover that's trying to do exactly that. Are there researchers working to understand and learn to enhance the transmissibility of viruses? Well, yes, they want to know what nature might throw at us next. How about evading the immune system more effectively? Well, if you're doing gene therapy and you want viral vectors, then yeah, that's a useful trait. And if it turns out it can be genetically encoded, then you could put it in the virus if it's a general purpose mechanism. Are there folks developing other ways of building invasively exponential agents? Well, I work in the field of gene drive. The answer is very clearly yes. So even if we can't reliably do it today, the odds are we will learn how to build invasively exponential agents that are in a reliable manner. And we should not assume that a sophisticated bioweapon would cause harm anything like the way that nature does it. Because if nature causes us harm, it's an accident. Evolution selects for replication, for replicative fitness, not for harm to the host. Harm to the host is only selected for if it enhances replication, which for most viruses and other pathogens, it does not. In fact, often, though not always, the opposite. We have much to learn here, but the point is we should be humble and assume that the odds are pretty good that we will be able to do this, or at least the odds are not so trivial that we can safely ignore it. And if we can learn how to do this, then we could plausibly learn how to weaponize this. So what can we do? Well, let's go back to our slow, obvious, and easily blocked. If we want to defeat pandemics specifically, a problem with the fast ones is that they're fast, but the fast ones tend to be respiratory. And there's a lot of talk, oh, well, if everyone had an N95, we'd be better off. Well, maybe. N95s aren't perfect. Aerosolized transmission can still get you if you hang around indoors, even wearing an N95, because it's called an N95 for a reason. It's not called an N100. But we do make the equivalent of N100s, and they're called powered air purifying respirators. They cost about $1,000 today. If we were to place an order for hundreds of millions of them, I'm sure we could get this cost down to $100 and make them much more comfortable to boot. So $35 billion for the United States to equip every citizen with a powered air purifying respirator with a bunch of spare filters could almost certainly be done. Do that, and we're done with respiratory pandemics, at least in wealthy nations. What about subtle things? Even if you take the fast things off the table, someone putting on their black hat could say, all right, well, it doesn't have to be fast. It just has to get to all of you. And if you don't notice it, you're not going to be able to defend yourselves. But if it's biological, it's made of nucleic acids. If it's made of nucleic acids, we can sequence the genome. If we were to sequence all wastewater in every major town and city, if we were to sequence natural waterways to pick up the sorts of exotic stuff like gene drives that could spread out there, and in places where that's possible, given health and healthcare systems and privacy laws, also clinical samples, then we could detect anything exhibiting exponential growth over an arbitrary time period just by looking at the time series and which sequence fragments are unique, or at least uniquely displaying an exponential growth pattern. Identify a novel threat, no matter what it looks like, right? Even if we come up with a universal coronavirus vaccine, for example, then that would work against natural coronaviruses, but it wouldn't work against an engineered one because a human would know how the vaccine works in advance. Evolution doesn't do that. An observatory could pick it up no matter what it looks like. And that's why sequencing is so critical. But the point is that even if we were to do both of these, we couldn't protect everyone. These are primarily defenses that could help the rich world. And you'll note that I didn't put anything there about blockable, because I haven't seen any great ideas for how to make everything blockable. The point is, if we develop something that enhances rich world defenses that also increases the risk of misuse, that seems ethically dubious because we are putting people at risk who will not be helped by our research if successful. Next, the challenge with this is if we are primarily protected today by our ignorance, that is, we don't actually know how to reliably build these things, or at least the tens of thousands of people with skill in mammalian tissue culture don't know how to build viruses that are going to actually cause the next pandemic. That is plausibly the case because people who might know are keeping it to themselves. After all, I must hold myself morally responsible for whatever comes of CRISPR-based gene drive. You probably don't want to be the one who discloses how to make the next pandemic, or worse. But that means inherently that we can all safely assume that it's actually worse than any of us thinks, that other people have thought of potentially catastrophic biorisks that we have not considered, and that they have wisely kept it to themselves. And that's plausibly a good thing because a naive person attempting to warn the world is a very plausible path to disaster. I'm concerned by the incentives loop. Directing evolution is all about considering what are the incentives on the relevant replicating nucleic acid molecule. Well, here, the replicating agent is not a molecule itself, it's the information, the instructions for making that molecule that can prove invasively exponential. And information can also be invasively exponential. After all, you just need the New York Times to write about it, and just about everybody relevant knows. That's fast. So suppose someone holds a competition, say. They want to identify novel biothreats so that we can start working on defenses. Well, first of all, this presumes that defenses are possible at our current technology level, which is a dangerous assumption. But suppose they do this anyway, or they're not thinking about it that way, or they are confident in their ability. But suppose the winning submission leaks and becomes publicly known. Well, it might be overlooked, but there are certainly journalists who specialize in this, and they might write about it. And certainly there are biosecurity researchers, scholars who specialize in biosecurity, who are often called to justify their existence, even in the age of the pandemic, which is a little bit shocking, but then a lot of people say, oh, well, that's a natural pandemic and nature's the greatest bioterrorist. Humans could never do anything like that, right? Well, they would be incentivized to point to it as a potential threat. And once you get enough biosecurity scholars and journalists writing about it, it starts becoming more broadly interesting. One might even say glamorous. And as soon as a scientific concept becomes glamorous, there is an immediate incentive for every bench researcher to explore it, because if it turns out it is in fact glamorous and interesting, then they could get a publication in Nature or Science or Cell. And if they do that, and it turns out that the weapon would in fact function the way it was postulated by the concerned person who first tried to warn the world, well, then anyone who could make that thing from synthetic DNA could do so and release it. And judging by the pandemic outside today, we would not be able to defend ourselves. So this seems like a heinously bad idea. There are very strong reasons why we might want to do that for some classes of ideas. But if we think that something like the current pandemic that we cannot defend against might result, then it seems pretty clearly a bad idea. So what can we do about this? Well, roughly we might say misuse is a function of the number with skill. And by the way, there's 300,000 people with a PhD in the life sciences. Even if only a small fraction of those could actually make and use something nasty, that's still a lot of people. And we know that in the 1990s, Aum Shinrikyo actively sought samples of Ebola virus to use as a bioweapon, and their lead scientist was a graduate researcher in virology who specialized in genetic engineering, which thankfully we couldn't really do very well in 1990. Matters are different today. So it's safe to assume that if lots of people can do it, someone's gonna snap for whatever reason, harmful, destructive ideology, mental health issues, whatever. So it's the number with skill and access and individual odds of misuse. So we really can't do anything about the number with skill. There's just a lot of biotech folks out there, and biotech does a lot of good. I'm sure a lot of us have great ambitions for what they hope biotech will achieve in our lifetimes. Nothing we can do about that. Odds of misuse, we could plausibly do something about mental health. I mean, I will point out that rates of depression among graduate students are double those of the general population. So actually providing support in cultural change and cautious awareness raising might be helpful there. And there are folks working on that. Access is the other one, because you not only need to have the skill, you need to have access to the relevant reagents. And while you can always get, a basic biosafety hood, incubator, et cetera, to do mammalian tissue culture in your garage for fairly cheap, you don't necessarily have access to, for example, the virus that you would need. So if we made sure that we weren't just shipping virus to anyone who wanted it on one side, made sure that instead of publishing protocols telling all scientists how to make viruses from scratch, no matter what they are, we instead set up a supply center that would make a custom virus for any lab who wanted it and did not disseminate the knowledge of how to easily do that. That seems like we could actually accelerate the research while limiting access. And the other way, of course, is DNA synthesis screening. We really need to make this universal, because in order to make a virus from scratch, you need to access the DNA. If you can't get a physical sample of something similar and then edit it, which is a pain, the easiest way by far is to order synthetic DNA and boot it up in tissue culture. Thanks to some really foresighted people in industry, we're now screening about 80% of synthetic DNA orders for gene-linked stuff. I'm not going to say what the cutoff is. But this is threatened for two reasons. Number one, synthesis is getting cheaper, but screening methods require humans, and that gets more expensive over time. And it's also threatened by the nascent rise of benchtop synthesis and assembly machines, right? Instead of placing your order to Twist, for example, or IDT, or GenScript, or one of the other companies, and getting it in next day mail, instead you would just punch out the order on your benchtop machine and it would spit out the relevant DNA. How can we do something about this? Well, this is a project that seeks to simultaneously address this issue of information hazards in the life sciences and access to DNA synthesis. So here's the problem. Current BLAST-based search is a similarity search algorithm. It says, okay, here's your hazard in the middle. This is the hazardous sequence. This is the genome of the nasty thing that we're worried about. And if you're doing a similarity search, you're going to catch all adjacent sequences in sequence space. And this is going to include a lot of unrelated sequences that just happen to be similar. And so these require humans to look at them and say, is this actually threatening or not? What we might do instead is take that sequence, identify snippets of it that seem important, screen for those exact matches to those important sequences. But then you could say, well, sure, but an adversary could just deliberately introduce mutations around the genome that are plausibly harmless. Well, sure they could, but if we take our signatures, our peptide fragments from the proteins of this thing that we want to guard against, and we predict a bunch of functional variants that could plausibly be those substitutions the adversary might choose, and we do this for some number of them throughout the genome, then we're doing the equivalent of screening for these pieces of that overall space without having to screen the whole space. And that means we can screen out sequences present in repositories, because again, we're searching for exact matches, not things that are similar. So by predicting and curating these functional variants, we get this random adversarial threshold search. The adversary doesn't know which ones we picked or how many variants we included. They just have to guess, and that means they have to include lots of mutations throughout the sequence. So how can we conceptualize this? Well, obviously we can screen for the whole wild type sequence. If you divide the wild type genome into fragment 1, fragment 2, fragment 3, fragment 4, all the way across the genome, we can easily search for all those. That's not very many. There's only less than 10 to the 9th base pairs of all viral sequences in the reference viral database today. But we can also pick for some of them and predict variants using current software programs for doing this, and include them in a database and look for those as well. And so if you have variants by increasing fitness costs, so red is something that's basically non-functional, very, very costly. Green is something more closer to wild type. We can pick a bunch of those to defend. And an adversary, someone who wants to make a pandemic virus, in order to defeat this, they have to pick one variant for every sequence all the way across if they want that functional hazard. And if they pick something that we've already chosen, like these two here on the right, then we detect that that's an order, and we can say no. And it's also plausible that they might pick something that is unknowingly breaks the function of their thing. And so if they were to escape screening and make it, it would just no longer work. So is this actually feasible? So we decided to treat this M13 bacteriophage as our test virus. It's actually harmless and infects E. coli. But we just decided, okay, we're going to predict the fitness of variants throughout the genome, and we built databases of varying sizes to test the fence. And then we had other folks put on their black hats and design 22,000 variants of these particular windows for each of the 9 windows. And then we tested them in the laboratory and actually measured their fitness values. So in other words, we actually did a red team exercise to see if our system would work. And what we found is this is 21,000 attacks, the rest were controls, on just one of those windows. This is one of the more defensible ones. But this is just 1 million database entries, which, I mean, we can afford a database of much larger than this. And what we found is that the vast majority of attacks that were functional, that is, the virus is fit enough to be transmitted human to human in the case of a human virus, were detected. Typically over 90% were detected, even if you draw the boundary line at measles. And this is important because measles is the most transmissible virus known. It has an R of 18. Every patient, on average, infects 18 additional people in a vulnerable population. So if we can build something that has a fitness cost that is more than 18-fold, that is 0.055, which is about here, then anything below this just would not work. And this is just one window. There is over 6,000 windows in the genome of this particular virus. And we can almost completely defeat even an adversary with perfect knowledge of fitness with just one. So this seems promising. It's very preliminary, but it seems promising. So how would this work? Take your hazards, fragment them into 57 base pair pieces, 19 amino acids, translate them, choose which particular fragments, which windows you want at random, compute the functional equivalents, remove any of them that raise false alarms, and then you have your database of signatures of that hazard. Then we don't want to tell people necessarily which ones we're screening for. We don't even necessarily want people to know what we think is a hazard. So we can use a form of distributed oblivious computation to essentially hash all of those to make this database. And then on the orders side, you have synthesis orders that basically get fragmented, translated, hashed, and then you can just directly compare it. See if they match. So this is a plausibly new way of doing synthesis screening that won't generate any false alarms and will allow us to actually disguise with what it is that we consider hazardous. We could screen DNA for things that scientists who are concerned warn us about without actually having to disclose what they are to anyone, even governments. And we can do it without interfering with research. And I don't want to get into the details here, but the basic idea is most labs already need institutional biosafety committee approval of what they're doing, which typically involves your list of genes and organisms. We can take those lists, scrape them for relevant IDs of GenBank sequences, and make an exemption list for that particular lab, and automatically compare. They can submit it with their order, with a certificate, and the system automatically checks to see if anything they're ordering that would otherwise hit the database also matches the exemption list, and if it does, they let it through. So no research delays, and it nudges research away from things that are thought to be hazardous without highlighting them, and it provides this info hazard safe action. Instead of warning the world, you could approach secure DNA authorities and guard against synthesis of those hazards without highlighting them. I just wanted to thank all the many, many people who are on this secure DNA project who have really made this happen, particularly our cryptographer colleagues, because I am utterly hopeless in that.
And thank you so much, Kevin, for your talk and for your time.
Thank you, Harshi.